Let me preface my remarks by just introducing us, the Allen Institute, because we're very different. We're sort of a very different animal from sort of academic or government organizations. So we are an independent, not-for-profit institute. We started in 2003. We do basic research in the neurosciences. We don't give out grants. Sorry, we burn a lot of money. We are still in our growing phase. When I came, we were 160. We're now 330. We're going to 500 people in a couple of years from now. We are really somewhere in a culture between a university and a startup. We don't have independent PIs. We focus on a few very large projects that can be done at scale and that require collaboration among very different disciplines, and then we execute. We're now in the second 10-year plan, so we have a 10-year runway. Well, actually, now it's reduced to 6 years. We have a 6-year runway left, and it's roughly $1 billion over this time. And so this 10-year plan is really to build, to understand comprehensively in mouse and in human cortex, a tiny bit of matter, roughly 1 by 1 by 1 millimeter, to understand all the constituent elements in that, particularly all the different cell types, to understand all the different types of synapses, to understand their development, to understand the neuroanatomy, to understand it, at least in mice, in behavior in the context of a behavior, and then to model all of that at a variety of scales, from very fine, detailed biophysical model to much more abstract point or population models. We just moved into a new building that Paul Allen has given us a couple of months ago. It's a gorgeous new building, all open space. And so we're all together, 300 of us in a couple of years, 500 of us scientists, engineers, and other specialists focusing on brain. So this is our donor, Paul Allen. The vast majority of our funding comes from Paul Allen, who really wants to understand how the brain works. This is not about making any money. We have right now no patents. We have everything we put online. So we are about big science, not at the scale of LHC, but certainly in the scale of biology or neuroscience, where a typical lab is 10 or 15 people. We assemble a large team, and then open science, in the sense that everything we do, we put out for anybody to download freely. And this is not in theory, this is not in the future, but this is actually, we've been doing this since more than 10 years, where all the data that we produce is publicly accessible via API once they pass internal quality control. You don't have to get a login. You can download everything we do without any commercial restriction, and all the data are accessible typically 2 to 3 years before the associated papers come out in Nature or Nature Neuroscience. That's where almost all of our papers have come out. Somewhat similar to what Karl-Heinz showed you, we also have a detailed 10-year plan, but if we really reduce it, since we are sort of more in a startup, rather than giving you a book, we have a one-page chart that we give everybody to put in their wallet. So we all know where we are and where we have to go. And this is what we promised Paul. This is what we promised our external advisory boards. We have external advisors coming in, checking us out so that we remain on track. We have product releases 3 times a year. We just had one last Thursday, and we work very, very, very hard, just like in a tech startup, in order to meet our deadlines for our data releases. So we're doing all sorts of things for the community. Particularly, we're pushing hard, working with many in the community to build standard protocols so we can all exchange data called Neurodata Without Borders. So whether it's EFIS data or photon calcium imaging data, we can all use the same data and the same metadata we've given out close to 20,000 transgenic mice now, hardware, special purpose electrodes, etc. So we studied the brains of 2 animals, mouse and human. They differ by a factor of pretty close to 1,000 in terms of mass, in terms of size, in terms of volume, in terms of area, in terms of number of neurons. We are focused on neocortex, which is, of course, only part of the brain. We focus on neocortex because it's a very flexible 2+epsilon dimensional computational tissue. In us, it's roughly as big as this, like a pizza, 14-inch. 2 to 3 millimeters thick. You have 2 of them highly rolled up and put inside your skull. And if you look in other animals like whales, where of course the tissue is much bigger, the width is more or less the same. Or you look at the smaller animals like a mouse, again it's 1,000 times smaller. It's a square centimeters, but again the thickness is roughly the same. It varies between 0.9 and 3 millimeter. It's also relatively homogeneous across cortex. Of course, there are many differences locally. And so the belief is that to first order, it's translational invariant. To first order, despite all the differences that people know of, it's translational invariant. There's some uniform computation that's being performed in a part of the brain that people typically refer to as a column. And that doesn't have to be anal about the column. It constitutes a useful working hypothesis that there's sort of an elemental canonical and computation that's being performed in this column. And we are really trying to understand it from a basic biological point of view. Here, it's remarkably similar. One of these tissues, this is part of cortex. One of them is human. The other one is mouse. It takes an expert armed with a microscope or a scale bar in order to say which one is which. So I always stress the great similarities between human and the mouse brain. Of course, there are differences. People always ask what makes humans special. We are as special as any other species is, right? We have specialization, so does any other species. We don't know there are many differences. We don't know the difference that makes the difference between us and mice. The one relevant difference that we all know that people typically acknowledge is the size difference. So our brain is, as I said, in cortex there's 16 billion neurons versus 14 million in a mouse. It's roughly a square centimeter in terms of volume, and it's roughly 1,200 square centimeters in area in human. That's, I think, the biggest difference. So to look at the similarity of the architecture of cortex, particularly the transcriptional architecture, we released this a couple of years ago. We did large scale in 1,000 locations in each of 6 different brains. We did microarray technology. This is a lot of data. And it took us 4 years on a highly standardized condition. And the output of this is summarized here. You can this is a really very useful visual tool, and you can go to our website and download it. What this shows you, any 2 places, there are 290 different places in the brain, and you can look what's the difference in terms of genes that are expressed, that are transcribed between this place and this place, or between this place and this place. And here it's expressed in a log space, in a color space. So blue, there are very few differences above threshold. And red, there's large difference. So what you see are the 3 relatively homogeneous areas, particularly down here, that's the cerebellum. So of course you all know the vast majority of neurons in your brain are here in the cerebellum. Of your 86 billion neurons, 69 billion are in the cerebellum. And they're really very, very similar to each other. Even in cortex, neurons transcriptional described are relatively similar. There are of course lots of differences, but they're much more similar to each other than to any other compared to any other part of the brain. These genes are very consistently expressed. So this is the expression because we use exactly this. We have the standard operating procedure where we do exactly the same thing over and over. So we do this in 6 brains, and you can see this is one particular gene, and you can see the pattern. It's expressed in these different brain areas in 6 different brains. 1 female, 2 African Americans. So they're different ages, different genders, different ethnicity. But you can see there is, at least among a very large fraction of the gene, a great deal of homogeneity from one brain to the next. Now, interesting, if you do PCA, if you just focus on the transcriptional architecture at the level of cortex, you do PCA, you can roughly explain half of the variance by space. In other words, if you unfold the cortical sheet, this pizza, I can put it down, and you ask, what's the difference in terms of genes expressed between any 2 places? Half of that difference is given by distance. The farther 2 places, the 2 places where you sample your cells are apart, and the larger the difference. So it really tells you something very physical about this tissue, right? And close by, it's more similar than far away. So this great these sort of results lead us to expect, once again, that there is something very homogeneous about cortex, although acknowledging that there will be these gradients that are ultimately going to be expressed gradients in receptors in various proteins. So our biggest project is called Project Mindscope, which probably has right now 250 people focused on, where we just look in a standard lab mice, C56. animals, they're all 56-day-old, where we look at the visual system. We're focusing on the visual system, not because it's a particular good model of the human visual system. It's not. It doesn't have a fovea. It doesn't move its eyes nearly as much as we do. But it's a perfectly decent piece of cortex. And we really understand vision reasonably well. It's probably the best understood modality. So there are roughly 45,000 neurons that come up from the eye, go through an intermediate relay in the LGN, and then end up in cortex. There are roughly 1 million neurons in various visual cortical areas, 1/3 of a million in primary visual cortex. Now, we do everything, by the way, we define this, and we've refined this over the last 12 years, a series of ever more accurate atlases. So now our latest atlas is a 3D volumetric atlas where we have pixels, 10 micrometer pixels, where we can locate any neuron that we record, whether it's using electrodes, using optical physiology, doing anatomy. We always can place within this 3D framework with high degree of accuracy. This has, at this voxel level, it has 1/3 of a billion voxels, the entire mouse brain. So we have these large data production pipeline. So we have one, and I'll briefly talk about these. We have one pipeline just for generating high-resolution connectivity data, with another pipeline for generating quite reliable slice data in the slice of the individual neurons that we want to understand, characterize. And then we're building these brain observatories. We'll release the first data in 3 months from now, on June 3rd, where we image and make available cellular-based data within a very high-resolution framework, within both a structural and a functional framework. I'll show that to you. Now, what connects all of these is that they all use the same animals. It's visual cortex of young adult C57 black 6J mice. All the data is mapped, as I said, to this common coordinate atlas that we got by averaging data from 1,675 animals, so it's very precise. And we all use common Cree lines. In other words, we all use common transgenic lines, and we make these transgenic lines available to the community. So that's a great advantage. So we know then, if we are talking about, let's say, a layer 2-3 line that expresses fluorescent labels in layer 2-3 pyramidal cells in visual cortex, we make that available to other researchers, and we're all talking about exactly the same animal. It really leads to a high standardization. And all of this data goes into a highly centralized and standardized LIMS. A database. All right. So we've done this now in several thousand mice, where we inject a virus with a fluorescent tracer that carries specific promoters into a specific part of the brain. We have 6 trained neurosurgeons that do this day in, day out, so they become very, very good at it. And then we slice it, then we wait 2 weeks, and we slice the brain in 140 slices, and we scan in. We have 6 of these machines running in parallel, where we scan in high-resolution 2-photon tomography, essentially, in 3D, and then where we informationally reconstruct them and make them available. So the first version of this that had 300 brains came out a couple of years ago. And of course, this is not the raw data. You have access to the raw data as well as processed data. This is, of course, not the raw data because I couldn't show it on my Mac. But you can really follow now these neurons very specifically throughout the entire brain, right? So we inject here and in the mouse brain and then reconstruct it through the entire brain. So this gives us, for example, a connectivity matrix. This is a connectivity matrix of 300 animals. So think each row going across here is one animal where we made one injection, let's say, in the medulla or in the pons or in the cortex. And then we look, and then you can see the going down the rows is where does this projection go to? Again, it's a logarithmic scale here. It's a logarithmic color scale. On the left, on the left side, you have the right hemisphere. So we always inject in the right hemisphere. So you see where it projects to in this hemisphere, and on the other side of the slide, you see the targets on the contralateral hemisphere. So it gives you a complete picture you can think of this as a crude version of Wij. It's not at the single neuron level, but it's at the level of mesoscopic discrete populations, at the mesoscopic level. Right, and so you can see, unlike, for example, social networks, it's a very densely connected. Typically, 30 to 40% of all the entries have entries different from 0, but the amplitude of the connectivity weights vary greatly. They're log normal distributed with probably across 5 orders of magnitude. So it's not just that an area projects or doesn't project. This, of course, makes life for modelers and theoreticians very difficult, but this is very typical of brains. So for instance, for visual cortex, where we are focusing on, there's a total input, different areas, 42 different areas that project into primary visual cortex, and at least 34 output projection to 34 different areas. That's the brain. Now, the other complexity of real brains compared to neural networks is there are many, many different cell types, right? So we realized roughly 200 years ago that all biological organisms consist of cells. And then we realized, okay, they're heart cells and kidney cells and skin cells, and now we know they're brain cells. And then, of course, neuroanatomists, starting with the late 19th century, and in combination with the dye industry, developed all these specific dyes to light up specific cells. So we know they're Purkinje cells, and they're photoreceptor cells, etc. So now, we know, for example, in the retina, just to do something that computationally you might think is very simple, i.e. converting a stream of photons into action potential, takes roughly 80 different cell types. And you understand nothing in modern neuroscience without referring to cell types. It's really absolutely essential to understand what type of neuron you're recording from, and to try to understand its specific role in a specific behavior and its specific role for specific computation. So how do you identify cell types? It has been done. The only place where it's been done pretty exhaustively is in the retina. It's not been done exhaustively in cortex. It's much more challenging in cortex. So we use a combination of different techniques. We use connectional properties. We use physiological properties. The three-dimensional morphology, both of the local dendrite and axon, as well as the distal axon, as well as the genes that are expressed. Right? And then we use that in combination with a variety of models. So we do electrophysiology on a standard condition in a slice. In other words, we take slice from visual cortex or LGN or higher order visual areas. And we, on a very standard condition, we have this pipeline, which is now manned by 20 people who do this day in, day out, recording either with 1 electrode or patching with 4 electrodes or with 8 electrodes in order to get synaptic connections among these neurons. And then we reconstruct neurons, or we have sort of the semi-automatic pipeline using a variety of different models, either sort of all active models that we do in combination with the Blue Brain Project and Henry Markham, then models with passive dendrites and active Hodgkin-Huxley conductance at the soma, and point models. Because we want to see, and then also we use population models. We'd like to understand what sort of models do you need in order to replicate what function. Because I suspect that although I spend most of my life, of course, doing detailed dendrites, that for many aspects of computation in the brain, you don't actually need dendrites. All right, then we have a variety of point neurons. You can download all of them, so not just one. We have LIVs and LIVs and GLIVs and GLM, a variety of different point models. And then here you can see, for example, this pipeline generates. These are some of the cells that are automatically generated. In black, you can see the experimental data. In red, the simulated data, both for spike trains as well as for individual spikes. And all of this is that you can, as I speak, you can download all of this data. This is Allen's cell type database. This shows you on the left the location. Most of them are in visual cortex, primary visual cortex. Of course, occasionally we also record from and from other areas. And at some point, we'd like, once we've satisfied ourselves that we've really completely understood the characteristics of cell types in V1, of course, then we want to replicate this in different areas to try to understand to what extent is it really the same scheme. Is there canonical cell type scheme that's replicated in V1 in all the other areas? I think there is with gradients. So I think the basic result will be the same, but then there will be these gradients that are superimposed. Anyway, so you can go to this website, you can click on an individual neuron, you know exactly where it is, you can click on it to get its morphology, you can click further to get its detailed shape, you can either get the individual images or you can get a three-dimensional reconstruction of its morphology. You can click here to get the individual data. All the trial runs have passed quality control. We reject a lot of neurons because they don't pass either morphological quality control or electrophysiological quality control. But once they pass, and all the data is made accessible in this very easy format. You can download all the data very easily. You can also download the various models in Hawk, if you want to do neuron simulations, or you can use for Python or Nest, you can download the single point model reconstruction of these neurons. So for each neuron, we have a variety of models available from very simple GLIFs, the simplest being just an integrated phi, to more complicated ones, to biophysical simple, to biophysical very complicated ones. Now, I'll show you some data. We have reason to believe there are roughly 50 different cell types, at least in primary visual cortex. And so in principle, that gives rise to 2,500 cell type specific connectivities, right, which we call W alpha beta, where alpha and beta is an index that runs across all the cell types. So the evidence seems to suggest that if you, let's say, record from a layer 5 thick-tufted pyramidal cell, they make one type of synapses onto another layer 5 thick-tufted pyramidal cell, but they make a different type of synapse that has a different plasticity or a different average synaptic weight distribution onto a thin-tufted layer 5 or onto a layer 5 inhibitory cell. And so we have to get all that data, which is, of course, expensive because it's a square. And then, of course, we also want to get the synaptic weight and synaptic properties of the afferent input, right? All the various thalamic areas, the subcortical areas, the other cortical areas, and the modulatory substances that modulate and influence the firing properties. And here by W alpha beta, what do I mean? I don't just mean the synaptic weight, the probability distribution of the synaptic weight as a function of the distance, and then also plasticity. We know, of course, that plasticity rules are very important, particularly short-term synaptic facilitation and synaptic depression. These are really critical to understand properties of a network, even in time courses as short as a couple of seconds, which is what we concentrate on. Again, we're focused on trying to understand specific behaviors, in our case, visual attention and visual object recognition over a 1 or 2 action perception cycle. In other words, we're trying to understand what happens inside the brain of the mouse as it runs along. There are various targets. I'll show you a movie. There are various targets. Sometimes it has to stop, sometimes not. We're trying to understand in detail what goes on under those conditions. We're using both structural data as well as electrophysiological data. So this I just mentioned, where we do sort of patching with 4-patch or with 8-patch to try to get this sort of data. This is the wonder of transgenic technology, which we can't do in humans, of course. But in mouse, you can do it. So you can label only, a subset of all the inhibitory cells in layer 2, 3. Or you can label the layer 1 neurogliaform cells. And then now you can put for example, fluorescent reporter in, and now you can visualize them. They glow green. And so now you can precisely target them with electrodes. So you're recording, for example, from 2 neurogliaform cells in layer 1. That's a wonder. And we really gotten very good at generating these in these mice. And then we're also using structural data. So we are part of the Micron program, the IARPA Micron program with Clay Reed and Nuno da Costa. So we're doing very detailed reconstruction, both array tomography as well as electron microscopy of a 1 by 1 by 1 millimeter, cubic millimeter of cortical tissue. So we're really trying to do every tool that we have available to really understand in detail to reconstruct this, right, where you cut every 40 nanometers. You got 25,000 cuts. You have to detail reconstruct them, and then you have to annotate that. This 1 by 1 by 1 piece of this speck of brain matter. And then at the same time with Stephen Smith, we're using array tomography where you cut it in 70 nanometer slices, but then you use antibodies, multiple generations of antibodies that you can wash off. So we actually know these are GABAergic synapses, these are glutamatergic synapses, these are noradrenergic synapses, so we can stain them and know the identity of individual synapses. Yeah, so we're setting up this farm now electro-microscopic platforms, so-called TEMCA. These are arrays, high-density cameras array with transmission electron microscopy. And then separately, we're setting up this array tomography. So here you can see this. So this allows you, here we image the complete millimeter column. This is a DAPI stain. You can see the individual cell bodies, and then we can zoom in closer and closer and closer. Now you can begin to see the antibody stains at the level of the individual synapses. So now here for the different colors, these are either excitatory or inhibitory. These are either pre or post-synaptic targets. And we have a large informational team that uses computer vision, and we work with the Neuromancer group at Google to try to take all this data and do semi-automated. I mean, the goal, of course, is to do completely automated, but that's still in the future right now, to do a semi-automatic reconstruction. So that in this 1 by 1 by 1 millimeter of tissue, we not only have first imaged it, so we know from a behaving animal what the receptor field properties of these neurons are, but then we cut it up and we know the detailed wiring using EM, and using this sort of antibody technique, we also know the different types of synapses. And we know from our previous analysis of cell types, how these different cell types, how they behave at least under slice condition. Yeah, and so as I mentioned before, we're doing this multi-patching to try to understand the detailed properties of synapses, both chemical synapses as well as gap, as well as electrical coupled synapses, and also to understand how these synaptic connectivity properties change as a function of distance between the two neurons that you record from. Currently, we believe the best way to characterize cell types is just to look at the genes that are expressed in their cells. You can now do this. There are now powerful techniques that allow you to do, finally, over the last 3 years, that allow you to do single-cell transcription analysis, where you sequence essentially the RNA that's exported from the nucleus into the cytoplasm for individual cells. Because ultimately what we want to do, for this one cell, we want to see which genes are expressed, which 10 or 20,000 RNA, well, typically we do 3 to 5 million to 10 million reads. We get the top 10,000 RNA species that are expressed. At the same time, in the same cell, we want to do the electrophysiology, and we want to reconstruct its morphology. So we can really understand how many, in these very high-dimensional spaces now, transcriptional space, electrophysiological space, and morphological space, how many different clusters are there? So here we did this just came out last month, where we did this purely using transcription analysis, where we found 49 different cell types. So this is in primary visual cortex, and this is sort of expected, and the order of magnitude is not surprising. There are 42, very satisfying for a nerd like me, there are 42 neuron types. So the answer is actually 42. We find there are 23 inhibitory ones, 23 GABAergic ones. They're really essentially clustered in 4 large families: somatostatin, VIP, and parvalbumin expressing neurons, and they express in different layers. And this is sort of all roughly compatible with what we know from other data. And then 19 different excitatory cell types that differ by the genes it expressed. In some of these, we've also have morphology. What we have not done, because it's so far, it really hasn't been possible to do this routinely, where you do both transcriptional and physiological and morphological analysis at the same neuron, and then do it at many hundreds or ultimately thousands of cells. So this has been 2,000 cells to try to do it in a very large number of cells, where you get the answer, where you can combine, yes, by both transcriptional and where they project and the way they look and the way they behave. They all behave differently, and there are indeed 42 different cell and different cell types. Now we're in the process of doing the same transcriptional technique in other brain areas, in other cortical areas, because we really want to know how different is this number in other areas. Now we're also doing this in humans. In humans, there are a few differences. So this is a neurosurgical tissue. We work now with 5 local area neurosurgeons, and any time now between once every week or once every 2 weeks, we get a piece of human brain. That's typically, like my pinky here, sometimes it's a sugar cube. 20 minutes ago, or half an hour ago, depending on traffic, this piece of brain, you can see there, was part of somebody's living brain and, maybe contained their memory of their first kiss. It's now sitting in our lab. Why? Because the patient had a deep tissue tumor. So here, can see where the yellow arrow points at. For example, epileptic seizure or tumor, those are the two pathologies we work with. And so to access that tissue, to remove it, the surgeon has to cut in his way. So the surgeon has to tunnel essentially in. And then most of this tissue is discarded. Some of it is given to pathologists, most of it is discarded. So we take advantage of this now, and then we can do exactly, well, not exactly the same, but we can do similar techniques than we can do in mouse. Of course, we don't have Cre lines, we don't have transgenic lines available, obviously, in humans. So we're now spending a lot of resources to develop very fast viral techniques that we can rapidly infect cells using, for example, herpes or other viruses, where we can infect the human cell with a particular promoter. So we know they carry a particular promoter, and we can differentiate them, and we can study the similarities or differences to these neurons in the mouse. Basically, the neurons, it's not surprising, they look similar and they behave very similar. In fact, turns out it's a really pleasant discovery we made. They're much easier to record. They're easier to record than mouse cells. They're larger, the cells, and more relevant, the slices last much longer, because I suspect because of lifetime, right? So a human, typically will live 80 years. The mouse will live 1 year in the wild, and so the human tissue is much more robust, and we can now routinely record 2 to 3 days from the human tissue and get physiology, to do morphological reconstruction, to do then three-dimensional reconstruction, and do the modeling. It's been challenging to get, because most of the tissue doesn't come from young mice, but comes from people who are 30, 40, 50, 60, 70, or 80 years old. So it's quite a challenge to extract the transcript, to do the RNA extraction. And so far now we're working with Illumina and the Craig Venter Institute to do mRNA from the nucleus, which is sort of its own protected environment, to also try to do transcriptional analysis. All right, then lastly, we're building, so my early education is in physics, and of course in physics we have these wonderful, like the TMT 30-meter telescopes, the LHC, we have these wonderful observatories where you have thousands of people working to build instruments. We don't have that yet in biology, partly because the instruments are still much cheaper than an instrument like a Hubble telescope or like a high-energy accelerator. Although that's changing as instrumentation becomes fancier and fancier, and you must do more and more controls in order to get highly reproducible, high-quality data of the sort that's difficult to obtain in academics labs, where typically you get a beautiful paper in Nature with one PDF, a flat PDF, and you have to believe it or not, and there really isn't any deep data that you can follow up and that you can mine, which of course also leads to the fact that 2 out of 3 biological experiments cannot be and can't be reproduced. So what we're trying to address that is to try to build these observatories where we take all the data, including all the metadata, try to do it again very reproducible, day in, day out the same, and make all the data and metadata available to the entire community. This helps us then to get feedback. People spend a lot of time criticizing and the data, both positively and negatively, which helps us to keep on improving the data. All right, so that's our paradigm. You just saw our paradigm there. So we have this training facility where we train our mice in these visual tasks. It's a one-dimensional degree of freedom, so they run on this disk, and they've been trained by getting water appropriately rewarded to do certain tasks, like, for example, recognize grading of a particular orientation and stop and lick to get some water, or otherwise run past it. And then, for instance, of course, we want to show that these parts of the brain that we study are actually causally involved. It's not just correlation like in an fMRI experiment of the sort I used to do in human, but we actually want to know that this area is actually causally involved. So again, it's the one of transgenic animals. We can put channel rhodopsin into, for example, the interneurons in V1. And then we can pulse a light on there, and to show on an individual trial that for that trial, if I turn off V1 for 1 second, the threshold of the animal shifts dramatically, or the behavior goes completely away. So we can causally show that various parts of the brain are actually involved in the task. Right? That's a fantastic advantage that these optogenetic tools have given us. Here, for example, we have a visual recognition task where we train the animal to make a discrimination. It got trained on 2 objects, either mushrooms or birds, at different scales and different orientation, and it can generalize even to orientations or scales that it hasn't previously seen. So it does what we would what in human psychophysics would be called translation or scale invariant object recognition. And now the question is, given that task, where are the neurons that are involved? We want to do that both throughout cortex as well as really focusing on primary visual cortex. We know, for example, so this is very similar to what you would do in a human. There are this bevy of visual areas. Note the scale here. So 1 by 1 by 1, 1 by 1 millimeter covers most of primary visual cortex. There are roughly 12 different visual areas compared to 36 in us. We identify them now in each mouse, and every mouse that goes into this observatory that we train, we know exactly where these functional areas are. Then we can do braid-wide recording. So we do both. We've got hours of this data, and it's really beautiful. For me, it's very tantalizing to put on some minimalist music or space music at night and then look at this. Well, because you can see, there's all sorts of things going on, and most of the time we don't have any idea what's going on. There are, for example, these big spikes, right? You see them there? Nobody in the field has any idea what they are, and so they just block them out and average over them because we don't know what they are. They don't seem to relate to heartbeat or to breathing or anything like that. It's some interesting signal, and so people, of course, typically what they do, they only focus on, let's say, primary visual cortex here, and they ignore everything else. But once you have this whole brain data, it's pretty cool. And now then we supplement that with very detailed data. So this is now data just looking in primary visual cortex. You can see the individual neurons. So of course we'd like to combine that to do the simultaneously whole brain imaging while also doing this very focused imaging. So we understand the context, at least at the mesoscopic scale, of the entire brain on a single trial, right? So this data and this previous data is all single trial data, right? So this is something totally impossible to do in humans, where you would have to average over many, many trials. This is a single trial of one animal running. And you can see, so they are recording from those specific locations. That's its running speed. There you can see the video. And this plus all the other metadata, the eye movement, the pupil size, all of that is going to be made available to everybody. All right, and then lastly, we're trying to make very quantitative models of this to really try to reproduce our own data, just like dog food, you eat your own dog food, to really try to reproduce our own data with our own models. And of course, to also put all of this stuff outside so other people can also reproduce it and hopefully or maybe even do better than we do. So here the challenge is that we don't just want to reproduce what you typically do where you have one experiment, you build a model to replicate that one experiment. But here the challenge is we have huge amount of data. So we have the structural data from EM and from array tomography. We have the mesoscopic data. So we know this, let's say V1, all the different areas it gets input to and then projects out. We have the cell type data, right? So we know what the different cell types that make up primary visual cortex, how they behave electrically speaking in their local morphology and their axons. And then we have the functional data that we get through tools like this. We're also supplementing that. I don't have time to show that. We're building, as part of this IMAC consortium with Howard Hughes Medical Institute, we're building these, we have built these very high density probes where we have up to 962 sites 20 micrometer apart to sample electrically up to 20 kilohertz. Because the trouble with the imaging data, it's at a very slow scale. It's either 7 hertz or 30 hertz. And of course, you don't see spikes, you see calcium signals. And so there is this link, we're patching at the same, in some other experiment with trying to understand quantitatively the link between the electrical activity and the calcium activity, but they're not one and the same. So that's the advantage you want to use also to combine that with electrophysiology, where you get very high temporal fidelity data, right, up to 20 kilohertz, so you can look at high-frequency oscillations and gamma and all of that, and synchrony. But the drawback of electrophysiology, you don't know anything about the genetic identity. That's the advantage of using optical transgenic animals where you put a GCAMP in a specific cell type. And so we want to reproduce all these different things and put them all together and to make sure that we really can say we understand this quantitatively. And so we get responses in the visual case. We have these various classical stimuli, the gratings, the isolated noise, bars, etc. Then we have natural images, and we also show natural movies to try to reconstruct now at different levels of resolution, from biophysical very detailed to biophysical less detailed with passive dendrites. We're also interested, of course, in computing the local field potential, have done so over the last years, then to point models, and then to statistical models, to try to understand what type of model do you need to answer what sort of question. In order, for example, to replicate this, do you need dendrites, yes or no? And so, so here this is a simulation just of layer, this sort of just an interim report here. This is a simulation of a layer 4 population of very detailed models, 50,000 of them. That's roughly all the cells in layer 4 in V1. The other good thing, we have all these numbers now. Right? We're not estimating, but we actually have we're beginning to get really good numbers of how many neurons are there in this particular brain of this particular animal. And so we can reproduce, these are the cellular responses in response to showing these images, and so we can reproduce that using different models. So that's work in progress. We'll also release that next year together with the entire simulation environment. So that's sort of an update. We work with a large number of people. It's very easy to work with us since we don't really have a lot of IP, etc., and other restriction. We work extensively with academia, both here and abroad. And because sort of part of our DNA is to make everything we do is to enable to further accelerate scientific discovery for the benefit of us all, including for the benefit ultimately of mankind. And that's some of our group. And I'd like to acknowledge Paul G. Allen and his sister Jody for funding all of this effort. And with that, I thank you for your attention.
That's a good section of mankind right there. Christof, your 10,000 cell layer 4 V1 simulation, or any of your simulations, how do you, not just the statistics of average probability of connectivity, but how precisely do you choose to connect the cells? And in particular, if in the real mouse or the real human, two stellates respond to a 30-degree bar, are they more likely to be connected? And how do you arrange that?
Yeah, so we now know that. I mean, there was physiological data, and we just Clay Rees just publishing a paper in Nature to show that if neurons are similar neurons of similar orientation, are more likely to be connected than if they're dissimilar, and also the synaptic size is larger if you're similar than if you're dissimilar orientation. We don't have learning right now, so we put in these we put in statistical properties, right, measuring this over and over again with statistical distribution, and we sample from those distribution. Ultimately, Paul, I think what you're probably driving at, we want ultimately a developmental program, and that's a totally separate effort we're doing to wire all the thing up and by itself, which right now we're not doing for this particular project. Because that's ultimately what you want, of course.
Yeah, so I'll talk to you about it next time we have a break. It's something that DARPA funded us to do for a few years, is exactly such a system, and it would be great to put it to use.
Hello, I have a question about dendrites. So you mentioned that in your thinking, there's some functions of the cortex that you can perform without dendrites, and for some you might actually need dendrites. What's your current thinking as to the delineation between those two groups of functions?
Well, it really depends. I mean, we know there's all sorts of, I did this for my PhD many years ago, probably before you were born. There's all sorts of cool things you can do in dendrites. The question is, what do they actually do in mouse visual cortex? There's some evidence that they're involved to help sharpen orientation selectivity. So one question is, do we need to put in dendrites, and is it passive, or do we even need to put in active dendrites to replicate some of the orientation selectivity?